I'm Laura Shannon. I'm an assistant professor in the Department of Horticultural Science. My lab studies potato breeding, genetics, and genomics. And the breeding program really functions in 3 different locations. We're housed on the St. Paul campus, where we have greenhouses and growth chambers that we do a lot of the very early crossing work, and that's also where most of our sort of basic science experimentation work kind of goes on. Then we have Grand Rapids as our early generation and seed increase site. And it's really important to have an early generation site that is distinct and different from where we do trials, because potatoes are clonal. And what that means is that to get a new potato, you plant a potato, right? And so your new potato has whatever diseases or pathogens the old potato had. So with potato, it is very important in our breeding program to keep our seed as disease-free as possible. And that is what we're doing at Grand Rapids. We're using Grand Rapids as an isolation site to grow seed that's as clean as possible. And then our trial site is in Becker, Minnesota. And we work in Becker, Minnesota because they have really sandy soils, and it is right around a lot of the potato growers in the state. So it's really the perfect environment to see how our new varieties do. So I'm going to talk a little bit about the potato breeding process. When you're planting potatoes, if you're planting potatoes in your garden, right, you are taking a tuber and planting it and getting a new plant out and more tubers out of that. And that's an exact clone of the parent of that tuber. But we're looking for new genetics, new varieties, and to do that, we can't have exact clones, right? We've got to mix it up. So in order to do that, we pick female parents we're interested in, we pick male parents we're interested in, we pollinate them, and we get berries on our potato plants. So potatoes are really closely related to tomatoes. So if you want to think about what this probably looks like, it looks a lot like tiny green tomatoes. That's what the berries for potatoes look like. So from that seed, which we call true potato seed, we take that seed and we turn it into mini tubers by growing it for a generation in the greenhouse. And we do this because mini tubers are just way easier to plant than true seed. And then we take the mini tubers out here to Grand Rapids, and we grow them out in single hills. So we grow one copy of each unique genetic individual in the field. Most of what we grow out of these in this first field year, right? Most of them don't even look like potatoes. They don't produce tubers. They produce tubers, but they're like weird shaped, or they're super long, or they're a color we don't like. So you just see this like incredible messy diversity that mostly isn't particularly useful to us at all. What we do with that is we go through, a group of us walk through the field, we pull them all up at the end of the year, we walk through the field, and we look at the tubers just out of the ground, and we generally pick about 1% of those. So we've been growing like 20,000 individuals out there, and we come out with a couple hundred individuals that we think look promising, that actually look like potatoes. So from this 1% selection in the first year, we're able to observe those genetic individuals more times, right? Because, so remember, they're clones, so we get to look at them again the next year. But this time, we've gone from a mini tuber to like a single plant worth of potatoes. So the next year, we're able to grow them in 12 hills. So we get to look at 12 copies of the same individual. So a big part of the process in general is ramping up the number we're looking at each time so that we get a better sense of what the variety will actually do when it's grown on a commercial scale. And so again, at the end of the year, we drive through the field, we pull them all up, and we pick out the ones that really look the best. But we also sort of get an idea about yield, and we get an idea about consistency. And so there's a year between each of these. So this year we've got a field year 1 in the field and a field year 2 in the field. And then we also have a field year 3 in the field. And this is the first time for us as a breeding program. And so that's when we move to Becker. So field year 3 is the first time we look at the plants in an environment that's pretty close to what we actually plan to grow them in. So we have them growing in Becker, and we have them growing in Becker for observation and to get a sense how they'll do in sandy soil, under irrigation, in the conditions we'd actually like to grow them in. And then we have them growing at Grand Rapids to get clean seed so we can grow them again in year 4. So we've only been a breeding program for 3 years, so we haven't done a year 4 yet. But what that will look like is we'll keep ramping up the scale and we'll keep winnowing down every year. So after the first year, we take about 1%. After the second year, we take about 25%. In the third year, we'll maybe take like 50% and move them on to the next cycle. So each year we will select a larger percentage, what we looked at, but we'll also keep bringing the number down and down and down, right? Until we winnow it to potatoes that will actually make promising varieties that growers might be interested in growing and people might be interested in eating. And that is our overall goal. So that is what we've got going on at Grand Rapids this year. And I really appreciate you watching this video and learning a little bit more about what we're doing.